Having a problem in constructing phylogenetic tree using NCBI and Mega? Don't you worry, because today I'm gonna show you how to look for evolutionary development and diversification of species. First, you need to have Mega downloaded, or the Molecular Evolutionary Genetics Analysis software. Once you have it, you can now go to NCBI website or the National Center for Biotechnology Information, and then make sure that it is set to all databases. Then you can now input the code that you have, and let's find out what happens when we search for it. And it showed us an information about Candida cellulolytica 26S ribosomal RNA gene partial sequence. And let's try to click this link to learn more information. And I can see here that U94928 is the accession number of the organism Candida cellulolytica. And this page shows us the taxonomy ID, scientific name, and the full lineage of the organism. And now let's go back to home page and we can see the PubMed. So this means that there is a written article about the organism. So if you want to read the article, you can click it and read the abstract. So you will have the idea about the article. And this is the DNA sequence of the organism Candida cellulolytica. So we don't need to copy it or type it. We can just click Fast A to send it over to Mega. So let's click Send to File, and always make sure that the format is in Fast A. Then let's create the file. Once the file is being created, you can click it and it will be loaded to Mega. So this Mega is loaded with Candida cellulolytica. But remember, our goal is to look for ancestors and how species are related to one another. So we need to load more relationships. So let's click Edit, Insert Sequence from File, and let's click this 3 species or sequences. And we can see here that it is almost aligned, right? So we can see here AAA, AACA, GAAA. So we need to align it, so our task here is to go further as to have all T's, all C's, all G's, and all A's. So we can click on the muscle alignment tool or choose alignment, but first do not forget to select all the sequences. So let's click alignment, align by muscle, and this will show you the computation or the presets of the software. So let's just click OK, and then it will be computed and it will be aligned. So don't worry about some of the misalignment. So because the software is not 100% accurate, but it is really helpful. So you can also see here that there are dashes. So this just only mean that the computer or the software cannot find what to be inputted in this blank. And once it is aligned, we can now export it. So let's export the alignment. Let's click Data and then click Export Alignment in FASTA format. And now let's choose a file name. So let's try to type sequence for phylogenetic tree construction, or PTC, and let's save it. Once it is saved, let's minimize this and let's make a phylogeny tree. So we will make, or we want to construct, a neighbor joining tree. And it wants to know which file will we choose. So remember what we have chosen. So we cannot see here the file even though that we are on the right folder. So make sure that you select all files, and we can see here sequence for PTC, and let's try to open it. And let's click OK. And protein coding nucleotide sequence data, we don't need that. No. Okay. So this is the sequence, right, of our phylogenetic tree. And one more time, let's try to create, construct, or test neighbor joining tree. And let's try to analyze. So these are the presets. Let's click OK. And now we have here the phylogenetic tree.